Yeah, okay. And now it is recording. Cool. Oh, actually we can poll. I didn't know that. Okay. So the same theme going over and over, and just to make a case in point today, I'm gonna, instead of starting the section with questions, I'm gonna explain something that perhaps it might have confused you before. It's related to one of the questions that we have worked on. And in that passage, it asks about what functional group in that compound could be important for the hydrolysis site reaction. So what I did first, I looked for the keywords. For one, is asking within compound 6. Remember that when they told you about something that is already given, read it very carefully. So it's not asking about something that is not present in compound 6. If there's a functional group that can do hydrolysis but is not in compound 6, you should eliminate that as soon as possible. And then secondly is asking about hydrolysis reaction. And when it came through that, the first thing that you should eliminate is the amine. Because if you think about it, hydrolysis, what is the nucleophile? The water is the nucleophile, right? And it happens that amine is also an electron abundant person and is looking for a nucleus to attack. So it happened that amine could also be a nucleophilic, doing a nucleophilic substitution. So you can't place two nucleophiles together because there will not be any reaction at all. So the first thing you should be eliminating is amine, right? And um, then going through the list that I mentioned, the nucleophilicity, then you have to think about what is your nucleophile? Is it weak? Is it neutral? Is it strong? Obviously, like water is not like a superb kind of a nucleophile. If you're still confused of what makes it a good nucleophile, go back to the previous section for OChem. um I did discuss it in one of the lesson. What makes a good nucleophile? Remember, something that is charged, a lot of electrons around, skinny, not bulky, suitable to like do a smooth attack. Those are good nucleophile. So, given that our water is not a very strong nucleophile, then the second thing that we can kind of come down to the line is what kind of solvent do we have? So obviously the leaving group is likely to be the person, well, not the person, I mean the functional group that water is going to attack. But looking at the solvent is it protic? Is it aprotic? Is it polar? Is it apolar? So all of these things we also have been discussing before. What kind of effect when it is protic does towards our nucleophile will be distracting. Some of them are likely to be interacting to be to the proton, the positive charge. When it get too busy, it doesn't get the chance to attack the leaving group, namely the electrophile, right? So in this case, we're having a methyl cyanide, which is not protic, it's a protic, and is also it is polar otherwise, but it's not something that would necessarily kind of allowing, because none of the functional group right here in the compound 6 is charged or like having a positive charge. So it's not likely that like right away that if you have like an acid to like protonate one of the species, then you can generate a positive charge. But in this case, it's not likely as well. So then we come down to the leaving group, which is all the functional group that is present in the compound 6. And you see a bunch of things, and again, I mentioned that amine should be your top choice to be eliminated. So just eliminate that since it is a nucleophile and water is a nucleophile in our kind of desired reaction for hydrolysis right here. Secondly, none of those leaving group is going to be charged. So that means that it's not going to be leaving very easily. Especially a solvent right now, it is a protein. Plus, we don't have acid to kind of act as the catalyst for this reaction. So that means that they're not going to be leaving very easily. So what comes down to is that the only thing that might have slightly positive charge is the polar is the carbonyl carbon bonded double bonded to the oxygen because of that polarity giving the electron being drawn towards the oxygen. It leaves the carbonyl carbon slightly positive. Otherwise, the other ones, you see what the other oxygen are bonding to. They're either bonding to a benzene. So, like benzene have a very stable pi stacking ring right there. Like it's just that cyclic functionality for the electron that is circulating right there is not gonna be dislocated to like pull any further electron from the oxygen. So it's not going to generate any positive charge on those oxygen, same as the one that is down below. So in that case, I would nail down to the one that has the slightly polar positive. But I do agree that this question is probably a little bit hard because it's not very obvious. Because even with the amide right there, we still need some sort of acid to catalyst to accelerate the reaction because we're gonna see it again in the reactivity. Amide for carbonyl reaction is not very reactive in a sense. Like if you're given other carbonyl compound, they are probably the least electrophilic. This slide I also presented to you guys last week and kind of named it as a opening question to you for you to think about what does it mean for electrophilicity is really the opposite of what it means by nucleophilicity. It just means that how easy is the leaving group gonna leave, you know? So what is gonna be good is gonna be something that has good leaving ability. The positive slash CORR stand for positive correlated. That means stronger the electrophilicity, meaning a stronger leaving ability. Think about what is gonna be very easy to leave. It's gonna be something that is very stable as an anion. So for something that is coming from the halogen group, those are gonna be very stable because they are kind of electron abundant in a sense, and they can hold the electron well. So also think about your solvent as well. Like what kind of solvent that they are gonna be in also kind of participate in that event. Because if you already gaining some sort of positive charge, for example, then it's gonna be making the electrophilicity easily to be attacked by the nucleophile. So the same thing that is applying right here. So now I'm gonna bring you guys to the first question of tonight. Um, Again, I wanted to test a little bit for you guys whether you have been paying attention since the very beginning of this series of sessions. I'm gonna give you guys probably just 1 minute because it's not complicated. Okay, are you guys done? This is something that I looked in like earlier this afternoon as I was trying to read through a bunch of people's lab thing. Um, so the first thing is, do you remember that whether you even need to read the passage? This is something that I have been emphasizing since Jan Chem era, which is historical, thinking back. Um, the first thing when you approach the questions, especially not in cause, like for cause you have to apply whatever that is indicated in the passage, but when you're doing other sections, there are gonna be individual questions that is encapsulated within the passage that you don't even need to rely on passage information. So here, I kind of asked, what kind of reaction is cytosine deamination, and what do you guys get? Hmm. Are you not sure um, for the questions? So for this one, yeah, it is A. It's a nucleophilic substitution. So think about what happened between from that amine group on the cytosine going to a carbonyl structure. It probably it has something that is attacking to the carbon that is bonded to the amine group, and then that amine is able to leave, right? And what is going to be attacking is likely to be something with the oxygen. So it happened that in this case, it is the water. So the second question came down from one of the students who asked me after class last time. It asked me that for something that I think I have seen it in the OChem, but I probably did not mention it specifically. Um, so it asked about aromaticity, and here is one of the questions that could be possible to be asked, and I give you guys 2 minutes to do these questions. So what do you guys get? I forgot to mention. So the one that is coming from before says uh, nucleophilic substitution is also a reaction for hydrolysis. So it's kind of going hand in hand for the example that I showed you guys. Do you guys need a little bit more time? Just wondering.
I do, just a little bit more.
Okay. All right. So okay. Let me see. So the first thing first is what defines aromaticity, right? So aromaticity meaning that you are engaging in a cyclic kind of structures, which both of them are, and they're flat. What does it mean by flat? Is the opportunity for them to become a sp2 structures. Remember that the sp2 geometry is a triangular planar structure. Not only that it allowed to form pi bonds, like up and above, then it with that pi bond in the sp2 flat cyclic structure is going to be allowing that little tunnel to go through up and down below the nucleus. So those two are essential. And the third thing is the Huckel's rule, which is stand for 4n plus 2. So if you a common kind of compound is like just like benzene. Benzene has like 3 double bond, and 3 double bond meaning is 3 times 2 electrons circulating in the pi bond that is connected, which is conjugated as well. So how does that apply right here? Like if I just show you the structure, which sometimes it happens that they don't show you the most direct structures that you might see. But just keep in mind that those those lone pair electron on the nitrogen can actually be participating in this cyclic structure with the conjugated pi bond. How is it gonna occur? It's gonna be requiring a little bit of your imagination and knowledge in resonance structure when the oxygen, the double bond oxygen, can pick up um, more electron holding the negative charge. It's gonna be allowing that nitrogen, which initially is amine, but it actually can form double bond. And when everyone is like engaging with those double bond, it's gonna be making the structure cyclic. So with this question specifically, actually both of them are aromatic. Okay? Okay. So then like I gave you an example of what is the aromaticity means and how this is gonna apply it. So the next thing is what is above structure? What is the name of that amino acid? And secondly, whether it is aromatic at all or not. Again, I'm gonna give you guys some time and just let me know when you guys are ready. So for this question, if since last time you guys did go over the amino acid structure, then that is great. But if you forgot, then that is the time to remind yourself to study it. Not only to study the structure, study the property, the 3 letter and 1 letter code, and also the full name. You just need to recognize that, okay? And here with histidine is aromatic because even though it is a 5-member ring, it can be changing. It can change its orientation in terms of the electron. And if you go back to one of the what's that called, the pyrimidine and the purine structure, you will see that um, histidine can carry some of the aromaticity. Let me see, is it right? One, two. Let me check back in that later. Okay. So the next thing is also the solvent again. Whether solvent helps by condition. Last time we kind of go through specifically targeting the nucleophile. When the nucleophile certain things, we put them in a certain solution. It helps them with the nucleophilic substitution. So here is a question from the textbook with the chapter question in the end, and just testing if you guys still remember that, okay? Alright, so nucleophilic substitution, although the name itself only talks about nucleophile, but we do know that nucleophilic substitution eventually the nucleophile to attack the electrophile, right? So important things to remember from previous lesson and this lesson again is that the basic solution makes the nucleus stronger. Uh, Again, we talked about why last time. Remember the charge? And secondly, the acidity is gonna be making the electrophile better. And why? And again, we did this question before. We used acid to add it into the reaction and making it faster. And then we were like, why? Because it makes the leave better, right? And why is it making it leaving better? Because it is transposing that positive charge so that the nucleophile can attack to it. Okay, so then we come down to certain again, I usually do not follow like book session topic by topic. Those are relatively straightforward. It's just, you got a nucleophile and then it's gonna attack to some sort of carbonyl carbon and then replacing whatever that is attaching and then forming back that double bond with the carbonyl carbon and then the oxygen on the top. So everything is really straightforward. Just a couple other things about heat, which I didn't kind of glide upon very much. The very last time when we talk about heat is more like thermodynamic and also the kinetic part. But this part is about how does it facilitate certain structure to form. So remember that heat kind of generate more collision. It's basically just like molecules swarming into each other and creating the heat. That is what you're actually feeling. So two of the things that is important right here. When I studied OChem, I studied beta decarboxylation. So I kind of count until I reach the beta carbon. But if you don't even know where is the beta carbon, then you just count. That is the best chance that you can get for decarboxylation, to be honest, because you can see that when in this figure that they're showing in the textbook, you can see that when the hydrogen is sticking out from the carboxylic acid, it's actually almost like a six-member ring. So that means that that is the most convenient distance for the double bonded carbon or double bonded oxygen to attack, leaving that to bond to get it to the hydrogen on the other end and allowing the carbon dioxide to leave. So that is what happened right there. Whereas on the other hand, another ring formation that is possible is either 5-member or 6-member ring, and that is also as well the most common structures. It happens in the glucose kind of sugar chemistry because they're stable. And when they're stable, then they can form. And remember that what makes them stable is some sort of strain that is being alleviated because they have involved more members. And what kind of strings that is involved? Or again, it is ring string, angle string that is confining how many members that they should be participating. So all these things is that we have talked before, it just mentioning it over and over again. And there's actually nothing new. And another thing, which the shows right here, the transesterification. So it's basically if you look at the reactant or like the left side or the right hand side, both of them basically comprise of either ethanol, which is on the left, or methanol, which is on the right. So both of them is like alcohol as well, and it's just a matter of chance of which direction is it going to. And again, it's like kind of jumping right and back, and if you jumping enough, then you reach equilibrium. I mean, if you have one equivalent on each of the side, then yes, you're in the middle. So you have half and half. But remember Le Chatelier's principles, which is again the same thing that we have been talking about. Which side I have more? Um, which side will be favored? Is there any heat that is generated particular to one side towards over the another one? So all of these things is just a kind of revision of what we have always been discussing. And then into the amino acid chemistry. Amino acid is there's not much about it in OChem at least. There's a little bit more when you go to biochem and learning about the secondary and tertiary and quaternary structure, but there is not much, to be honest. I think the most important thing is for you to remember that all of the amino acid is L, which is the optical direction that the polarity that when they shine the polarized light, which direction it is gonna rotate. Everyone is L except cysteine, which is D, and glycine, which is a chiral. Because glycine they have a hydrogen for the uh, R group, so that's why two hydrogen you can't make a chiral center out of that. And secondly, cysteine is just a kind of priority assignment that happens to have a S on one side, so allowing that to be the D amino acid because of the assignment of priority when you're going through that priority group for the alpha carbon. So, and secondly, the second important thing is going to be the peptide bond, which remember very, very carefully. I think it is not like not much to me at least. Like how I remember carbon like the peptide bond is the NCC, and that is how I kind of remember it. Like so for every amino acid it's gonna be having an N group and a C group, a carboxylic group. So I just count from NCC and that is one. And then NCC that is second amino acid. And then NCC, okay, that is the third amino acid. So every amino acid I just go through NCC, 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 NCC. And you can actually just tell by the direction, like which way is it going, whether the N terminal is on the left or N terminal is on the right, because everything is the same direction. So you cannot have something that is going like from the from this end right here going to the left, right? Because you see that this is the carbonyl carbon, and carbonyl carbon should be the third one. So you can only have NCC, NCC in this direction, but not like NCC in this little one. Because if when you go through the center, the middle one should have the R group with it, but not a carbonyl. Okay. And also the second thing, partial double bond, like that is the only thing that whenever that they ask me anything about peptide bond, the only thing that I remember, the partial double bond. And what does partial double bond mean? Like remember that I always derive everything by one single word because I don't like memorization. So partial double bond meaning that it can resonate, right? Because otherwise, how is it gonna make it double partial double, right? So the only way to make it partial double, meaning there's resonance structure that occur, and since there's double bond character, that means that it's gonna be maintaining some kind of planar structure. Remember sp2. sp2 hybridization is gonna maintain some of the polar uh polar plane that is confining the geometry. And with that, because you can think that a paper can only be a paper that is a kind of flat paper, and they cannot they cannot be like kind of twist it very much, because you're gonna, if you twisted the paper too much, think about how you twist the paper. When you twist the paper too much, the paper is gonna be torn into two pieces. So that means that to some extent it's gonna be confined by angle and rotational strain. Like those are the things that is confining, and therefore when you're reaching to the secondary structures for a peptide, is those are the factor that is coming into play. Why the peptide can only conform to like alpha helix or beta sheet. So those are the things that is explaining why only certain secondary structure can be adopted. I don't know why I feel like I'm being really quick, but I'm not sure whether I'm being really quick. So let me pause a moment if you guys have any questions. Okay, I'm gonna send out the slide for the question. Um, so I'm gonna wait until um 10 minutes and then come back again, read through the passage. Um, the full thing that to do, read the question really carefully, read the passage really carefully, and then be confident in your answer. Choose the one that you think it is most correct. And then the fourth thing is, if you see something that you don't know, don't panic. Come back afterwards. Mm. Are you guys ready? Okay, perfect. Okay. So this questions and kind of again stop me at any time if you do have questions. Um, so for this passage, it's pretty straightforward. Not a lot of reading. Like the ones that has a lot of reading kind of startled me because I'm not very good at it. So for this specific protease is talking about HIV protease. One of the things that I think I forgot to mention is the peptide bond. When you do a cleavage of peptide bond, it's usually very often a hydrolysis process. So I'm saying usually. That means there's exceptions. So just keep in mind that I think usually in on MCAT they will not gonna ask you other thing besides water. So it's talking about this example of HIV protease being a aspartyl protease, and then it explained blah 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 what is this aspartyl amylase does. Okay, and then I was like, cool. And then it says since it is a relatively small, so it's describing the structure of this protein. So it can be it's talking about the benefit of why it's a good drug. I'm like, okay, cool, but none of my matter that matters to me. Um, and then I go through, so they showed you an example of how the cleavage occur, and I'm like, okay. And then here it says that using the molecular model, the researchers designed and synthesizes the transition state analog. So I don't know if they mentioned in the biochem um, when there's a transition state, that means that it allows the most perfectly bind. It's the kind of state that has the greatest affinity between the enzyme and the substrate itself. That means that this protease cleavage site is probably very, very binding strongly. Well, at least it's the strongest moment to that protease. So given that it is a transition state analog meaning it's something that is fake, right? So it means that it's actually not the target of HIV protease. Um, it's just something that is kind of locked right there. And then here it talks about the compound one. Compound one has a half-life, and what is compound one? Compound one is the transition state analog, okay? And then it says that the figure 2 is gonna be one of the HIV, um, potent HIV protease inhibitor. Okay, so that means that this being a transition state analog is gonna inhibit the HIV protease. And then it describes its half-life, how is it metabolized, meaning how is it gonna break down. It's by this enzyme. I don't know if this enzyme is gonna do any relationship with our questions. I'm like, okay, noted. And then it talks about this major metabolite results from, then I highlight this because it's talking about this metabolization by the CYP3A. How is it going to occur? It says that it's by this dealkylation of the CH2R group. And I'm like, okay, yeah. And then the next paragraph says that one of the main side effects is the kidney stones. What's the kidney stone? Kidney stone is the calcium oxalate. And then I also highlight that and say noted. Yeah. And then so the researcher blah 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 blah. So they basically wanted to improve it so that they can use it better. And then I'm like, cool. So the first question asks about how does the compound one used to treat HIV infection. And if you, since we already break down the paragraph itself, and if you do see that they say that one of the compound 1 is a potent HIV protease inhibitor. That means that it is counteracting what HIV protease is trying to do, which is to cleave the protein, the peptide bond. So when something is acting against its intended action, that leaves us to the answer B, um, the antagonist. Let me get a little box. Okay, card free. Okay, okay, okay. Let me check. Okay, then let me copy and paste this. All right. So question 2 talk about the squalene. So one of the first thing that came into my mind is whether so the paragraph itself, the essay itself doesn't even talk about squalene. Where's this coming from? I'm like, sure. But I added in there for this purpose is that this is a question that coming from the AAMC practice exam, and it's shown as one of the discrete questions. So I thought that is good to kind of camouflage it into an essay and trying to see if you will spend 1,000 years on these questions. So if you look closely in the structure, remember something that I mentioned just now, um, with the previous slide, what happened with the cyclic rearrangement, usually by heat, it is possible allowing certain member ring structure to be formed. And what are those members? Those are the 5 or 6 member ring. And if you look closely in the squalene, then at one moment you're gonna realize that they are forming certain ring structure just without that last bond that is closing up the circle, the cyclic structure. And usually how the squalene does that is that they form this little ring form and then leaving the opening out. And then one of those things will come along and allowing the double bond, the electron on this double bond to move. So once they move to close up the gate of that cyclic structure, then it allows another one to move. Then this will close, and then this will close, and then this will close, and then this will close. I hope you can see my little arrow. Otherwise, it's gonna be very hard to imagine um, what I'm trying to say. So basically, this. So there's a 6-member ring, and then going right here, then there's a 5-member ring, and then a 6-member ring, and then a 6-member ring right here, and then 6-member ring right here. And if you did kind of review a little bit of the biochemistry compounds, it is one of the steroid hormones. Steroid hormone is known that it is made from cholesterol, and cholesterol is actually made from squalene, and squalene is made from terpenes, a bunch of terpenes. And terpenes is one of the things that is in the OChem that we're supposed to study. Is this little Y shape. I like it. It's Y shape. But even if you don't know what is you know, a squalene, just look at the structure. Like in terms of the naming, for answer A, it says is a triacylglycerol, and we don't see any oxygen right here, so that means that it's not going to be raw. And there's not even a triacyl, which is 3 individual acyl group right there. So A is out, phospholipid. We don't even see one single phosphorus right here. So it wouldn't be a lipid. Prostaglandin. Remember that I mentioned before that usually a signaling molecule. Something that end with IN, some like insulin, is usually gonna be a protein structure, and this obviously is not looking like a protein structure. So by elimination, it should also bring you down to the steroid hormone as your answer. Okay. So then the question number 3 is why do kidney stone form? And they give you a bunch of structure, a bunch of equation, and obviously they're trying to test you on your understanding of KSP. And in GenChem, I did mention that KSP is basically just one of the indicator of the equilibrium position, and usually SP is basically meaning when is the solid is going to be precipitate. And how you're gonna be able to measure that is basically multiplying, not addition. So we can eliminate A and C. So then it came down to B or D. B or D, one of them say is greater, one of them saying is smaller. And when something is greater than equilibrium, that means there's more concentration than the solution can hold without precipitating. So that means that then when the concentration is so high in that solution that it can hold, that means that it's going to form a solid as precipitate. That's why it's going to be answer choice B. And then the question 4, which I have prepared myself a little equation. So question 4 is D. So you have to basically think about how many times that you have to do reduction, how many hydrogen that you need to add upon this carbonyl group. So you can see that from this double bonded carbon to the oxygen, it's coming down, becoming an alcohol, and adding another hydrogen group. That means that you're going to need 2 hydrogen. So 1 and then 2 hydrogen. One is from the NADH, and then the second one is going to be the proton. And also, when you're having a proton, you can see that the charge is basically balanced with both sides becoming neutral. So, in terms of that, that is why it's making the answer. Basically, you just have to balance the equation. And that's all that you have to do for this. And again, this is a individual discrete questions. It does not require any of the knowledge from the article. So hopefully you can identify this. And then question 5 talks about the peptide bond that is being cleaved. That means that we have to go back to figure 1 where there's a cleavage site. And when we go through, remember NCC, NCC, NCC. So let's see. This is the N. Remember that is not going to the carbonyl carbon. It is going to the alpha carbon. So the next C is the alpha carbon. So NCC, NCC right here, NCC, and then okay, that's cool. So you can see immediately that you can see the phenyl group, and the only thing that has a phenyl group is the phenylalanine. Basically, it's the alanine with the phenyl group, right? So the left is the phenylalanine, and looking on the right is very distinctive N-cyclic structure connecting to the carbon alpha carbon. The alpha carbon. So this is a very signature of the proline. So proline with the phenylalanine is making the answer D. And then the last questions asked about, to me, should be the simplest questions. How many stereoisomers that compound 1 makes? So stereoisomers, compound one, and then you go to compound 1, and then you start counting. One of them is from here. Remember to eliminate anything that is double bonded because that is not a chiral. It has to have 4 different groups that is connecting the carbon. So here is one, and then the second one is this alcohol connected carbon, and then the third one, and the fourth one right here, and then the fifth one on this 5-member ring. So together is 5, 5, and then to do the math of doing the center, I don't know if I forgot to mention before, it is number of isomers that you can form is 2 to the power of n. N stands for n is the number of stereocenter, or no, center, I mean. Okay. That makes our answer to be 32. So that is C. Okay. Let me stop the recording.